This video is about the polymerase chain reaction, better known as PCR. Today we'll be going over how PCR works, applications, and future uses. PCR was invented by Dr. Kary Mullis in 1983. He discovered it when he noticed that Sanger sequencing was ineffective when concentrations of DNA was too low. PCR used to be slow and labor-intensive due to the manual transfers of test tubes between water baths, each one at a different temperature relating to one step of PCR. Eventually, PCR would use Taq polymerase, which comes from bacteria in hot springs, allowing it to work at high temperatures, removing the need to add DNA polymerase after each round. Cetus corporations developed a temperature cycler, meaning there was no more manual transfers between water baths. PCR requires template DNA, primers, nucleotides, DNTPs, and Taq polymerase. Primers designate which sequence is to be amplified on the template strand. Taq polymerase will attach to primers, adding on nucleotides. The materials listed are put into a test tube and put into a thermocycler with preset temperatures. Step 1 is denaturing. Around 94 degrees Celsius, the H-bonds break, causing double-stranded DNA to become single-stranded DNA. Step 2, annealing. Primers are complementary to the end regions of the desired sequence to be amplified. Step 3, extensions. Taq polymerase attaches to primers extending using complementary DNTPs. These steps are repeated, and every cycle exponentially increases the number of DNA molecules. Qualitative PCR determines the presence or absence of a certain gene. It can show if a pathogen is present, while quantitative PCR can detect and quantify the amount of a particular gene's presence. This is also known as qRT-PCR, which detects and quantifies the product of PCR in real time while being synthesized. PCR can be applied to many different fields. PCR is used for COVID-19 tests in laboratories for quick and effective diagnosis of the SARS-CoV-2 virus, reducing the spread of COVID. Food safety tests can be done to ensure food safety standards are held at a high degree, and tests can be completed within 2 hours compared to the typical 18-hour tests. Lastly, it helps solve sexual assault cases faster than ever by expanding the DNA sequence from victims so perpetrators can be caught sooner. PCR will be a forefront technique in next-gen sequencing, sequencing small parts of a genome rather than sequencing the entire genome, which is costly and slow. Extreme PCR and other breakthrough PCR methods will continue to improve the technique, solidifying its future in genetic analysis.
Created using Powtoon.